Okay, here we go. This is the functional groups video. Hopefully it's going to be a lot shorter than the identifying non-covalent interactions and how to draw them. These are the functional groups that you need to know by heart. And what I mean by heart is you need to know their names and structures. These functional groups are the ones that are biologically relevant, so knowing their names and their structures is just part of learning the language of biochemistry. You also need to know whether the functional group is polar, and if it is polar, is it neutral, positively charged, or negatively charged, or if it's nonpolar. And then I'm also going to introduce what we in biochemistry call CPK coloring. You may have been introduced to this in previous chemistry courses, but I want to bring it up again because it's going to be important for your molecular modeling assignment and because biochemistry is a very, very visual science and we tend to use certain colors to represent certain things. So let's get started. We're going to start with the CPK coloring first, and then we'll go through and we'll draw the different functional groups that you need to know by category. So I'm going to do the categories, nonpolar, polar neutral, polar positive, and polar negative. All right. So this, I don't know, what do we call this? Figure, graphic, whatever, is from a different biochemistry book, but I really like it a lot. If you could take, 5 seconds to pause the video, I want you to think about what do the colors mean here? So go ahead and pause the video, and I want you to figure out what do the individual colors mean. That whistling means that my laundry's done. Forgot it was going. Okay, so when I take a look at this, science is a lot about pattern recognition, and so what I want you to see is that these ones right here are all gray. They're all hydrocarbons, and in CPK coloring, carbon is black or gray, and hydrogen is white. And so if you have a hydrocarbon and carbon is black and hydrogen is white, you get what's like a gray color. Okay? All of these ones that are in red all contain oxygen. So in CPK coloring, oxygen is red. And I'm going to be circling the functional groups that you need to have memorized. I'll try and do this by color. Okay, nitrogen is blue. And the functional groups that contain nitrogen that you need to know, I will circle. Okay. Sulfur is yellow. And I don't have an orange color, so I'm just going to make my red a little bit opaque. But phosphorus is orange. All right, so this is CPK coloring. When you get to the molecular modeling assignment and look at just various figures in the textbook, oftentimes things are represented in what's called ball and stick. So, for example, you may see a single red ball that is connected to, a carbon, which is connected to another red one. And what you know there is that you're looking at some sort of carbonyl. Maybe it's an ester, maybe it's a carboxylic acid. You're not really sure because you can't see the rest of it. So the different colored atoms represent the different colors represent the different atoms. And it turns out that in the molecular modeling programs like Chimera or Pymol or Jmol, the only color that you can change, the only atom that you can change the color of is carbon. So if you see a protein that's colored pink or purple, The only atom that's actually been changed is the carbon. Everything else has to stay the same. So, food for thought. All right, let's get to drawing. Again, we're going to start with the nonpolars, then go to polar neutrals, and then finish with the polar positives and polar negatives. All right, let's go green. All right, so here are the nonpolar functional groups that you need to know by name and structure. We're going to start with the simple ones that you have probably learned first in organic chemistry. These are the methyl, ethyl, propyls, and butyls. These are the straight up alkane hydrocarbons. I'm going to draw them all out first. So all I'm doing is sequentially adding one more carbon each time. So the chain grows by one. I have them written in what's called condensed formula, not skeletal, not Lewis. All right, so the simplest one with just one carbon is methyl. That's an H. Adding a second carbon, you get ethyl. A third one is propyl. And the fourth one is butyl. Again, these are the alkanes. They could be alkenes and alkynes. You can add all sorts of other things to them. But what you'll notice is that they're all hydrocarbon. There's very little electronegativity difference between the carbon and the hydrogen, so that makes them nonpolar. The other two that I'm going to add to this, are the phenyl. Probably should put my little R groups on here, huh? R just represents anything else that is attached to this. Phenyl group is, you probably recognize it as a benzene ring, but we call it a phenyl. And then the other one is a phenol, which is the benzene ring plus an OH. So that's phenol. Okay, and those are the nonpolar. Sometimes the phenyl group is considered a polar neutral. In biochemistry, we're going to add it to the nonpolar category, just to make things simple. So those are the nonpolars that you need to know by name and by structure, and by know them I mean by heart. All right, moving on. Going to the polar neutrals. So these are molecules that have some sort of electronegativity difference, dipole moments, but are not charged. So they're not anions and they're not cations. So again, these are going to be the polar neutrals. Okay, there's a whole bunch of these. Let's see what I've got. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So space it out right. Again, I hope that you're taking notes as we're going. That would be the best way to use these videos, again, so you can use this in class on your homeworks and whatnot. Okay, I'm going to go a little bit skinnier with the writing utensil. All right, let's start with the simplest, which is the ketone. It's a carbon, also known as the carbonyl. So it's a carbon double bonded to an oxygen. Call it a ketone. It can also be called a carbonyl. Okay. The next series of these are just going to be derivations of the ketone, where we change the R group to a specific atom. So if we take one of the R groups and turn it into a hydrogen, it becomes an aldehyde. Okay. If I change that hydrogen to an oxygen and add another R group here, it's an ester. If I take the ester and change the oxygen into a sulfur, please remember this one, it's very important. We call it a thioester. Thio is the prefix for sulfur. If I take that sulfur atom and change it into a methyl group, this is called an acetyl group. Now, if you were identifying these, say, from a structure that was given, you would not circle the ketone separate from the methyl. This, because of the ketone, is connected to the methyl group. This entire thing is a very unique functional group. And I would say this is probably one of the most important functional groups in biochemistry. Food for thought. Okay. Another very important functional group is instead of the methyl group, we put an NH, and I'm going to put an R here. And I'm actually going to draw this two ways. So there's a carbonyl carbon to NH2. So instead of just the R, I replace the R with a second hydrogen. This is called an amido. Again, very, very, very important. Probably my favorite functional group. Very important functional group in biochemistry. Again, you wouldn't circle the NH2 separate from the ketone if you were trying to identify functional groups. Because the ketone, the carbonyl, is connected to the NH2, that makes it its own special functional group. So you would circle this as its own entity if you were be asked to circle functional groups from a structure. Okay, the rest of these that I'm going to draw off to the side over here because they're not derivations of the carbonyl or the ketone. All right, first up is the OH. This, I call it a hydroxyl. You may have also learned it as an alcohol. Either way is fine. Just so you know, I call it a hydroxyl. If you switch out that oxygen for sulfur to get an SH, that's a sulfahydryl. Sulfahydryl. You might need to check my spelling on that one. If you put two sulfahydryls together, we'll talk about this reaction just briefly in biochem. This is called a disulfide. Very important in protein structures. Di meaning two, sulfa meaning sulfur, so disulfide. All right, and then the last one, I'm going to use this whole space right here to show you how I draw what's called an imidazole. Imidazole. All right, I'm going to put the R group at the top, one bond coming down. Now I'm going to go in a circle, a 5-membered ring, going that would be counterclockwise. All right, so I'm going to put nitrogen. And it turns out in this 5-membered ring, there is a nitrogen every other atom as long as you go counterclockwise. So we go nitrogen, carbon, nitrogen, carbon. Okay? The first nitrogen gets a hydrogen. The second one gets a double bond. And I'm going to put my little lone pair there so you can see that. And there's always two double bonds in the imidazole, and that second double bond is right there. So again, I'll just draw it one more time. R coming down, go counterclockwise. Nitrogen is every other. Okay, so it goes nitrogen, carbon, and then you get two carbons in a row up there. The first nitrogen gets the hydrogen, the second one gets the double bond, and then to complete the structure, you have to add the second double bond in there. So that's the imidazole. So those are the polar neutrals. Let's see. I think the imidazole is probably the hardest one to remember and the hardest one to draw. But it's super important. It's one of the functional groups on one of the most important amino acids. So the sooner you learn that one, the better off you'll be later. Okay, then the last slide or piece here, I'm going to do the polar positives and the polar negatives all on the same one because there really aren't that many of them. So I'm going to have polar positives here and polar negatives here. Okay, and again, later we're going to talk about how you determine whether a functional group has a charge or not, but for now you can just use the charges that I'm giving you. These are all at pH 7. Same with the polar neutrals, those are all drawn at pH 7. All right, polar positives. A nitrogen with 4 bonds on it, 3 of them occupied by hydrogen. Makes that nitrogen have a positive charge. This is called an amino, not to be confused with the amido, which is one of the polar neutrals. Okay, the second polar positive is probably the second hardest functional group to remember. This is called a guanidinium. I'll spell it down here. guanidinium. Again, a really important functional group to remember because it's part of one of the amino acids. So we start with a nitrogen, going to a carbon, and then coming off of this carbon are 2 more nitrogens. Whoops. And at the end, there are more nitrogens. All right, the first nitrogen gets a hydrogen, same kind of with the imidazole. Over here on the end, one of these gets a double bond, 2 hydrogens, and is positively charged. The other one just gets the 2 hydrogens and is neutral. So overall, each of these polar positives has a +1 charge. Hopefully you can see that. And then for the polar negatives, there are 3 of them. The first one is the carboxylic acid. Carboxylic acid, also known as the carboxylate. Carboxylate. Just food for thought, the 8 and the ic acid are the same thing. So aspartate and aspartic acid are the same thing. Glutamate and glutamic acid are the same thing. So the ending 8 is the same as the ending ic acid. Okay. The second one is the phosphoryl group, which is a phosphate or a phosphorus attached to 4 oxygens, 2 negative charges there. I call this a phosphate group. You'll see it also as a phosphoryl. Okay. Oops, I keep doing that. And then the last one, which you may or may not be familiar with, I'm not sure if you are, is called a phosphoanhydride. And that is basically 2 or more phosphoryl groups connected. So I'll start with my R, O, P, O, P, O. And then again, remember that every phosphorus atom has to be connected to 4 oxygens. I'm going to add all these negative charges here. This is called phosphoanhydride. And it may look familiar to you because it is important functional group in ATP. So the TP is a triphosphate. What I have drawn here is a DP, a diphosphate. And the connection between the 2 phosphate groups is the phosphoanhydride bond. And that about wraps it up for all of the functional groups that you need to know in biochemistry and their classification as nonpolar, polar neutral, polar positive, and polar negative. Have fun, and I'll see you in class.